Hey everyone, sorry for the delays and super excited to be here. My name is Dinkar. I'm a machine learning engineer in PathAI, which is a Boston-based AI and biotech company. And today I'm going to talk about a new technique for domain generalization for medical imaging, specifically for microscopy images that we developed using PyTorch. So this talk is divided into two parts. In the first half, I will cover pathology images, which are one type of these images, why pathology matters in a patient journey, and the importance of domain generalization there. Second half, we'll go through the technique that we developed, which we call as ContriMix, some result on existing benchmarks, and how we used PyTorch in the process. So to begin, a pathology diagnosis is often the gold standard to determine the presence or severity of diseases. And let me give an example to demonstrate this point. So a typical patient journey starts with some symptoms, which is usually followed by some non-invasive tests like radiology. At this point, if there is some evidence of malignancy, then pathology is used to arrive at a definitive diagnosis. It involves extracting a piece of tissue called a biopsy that is analyzed by a pathologist. This process is quite subjective. It is a hard task, and the reasons for that we'll cover in the upcoming slides. So this figure serves to highlight the pathology tissue preparation. It is a complex procedure with many steps. First, a piece of tissue is embedded in a wax block. A thin section is cut off, put on a glass slide, and stained. This glass slide is used for microscopic examination of tissue. And this is how the data, that is the whole slide images, short form is WSI, look like. This one is from lymph node with metastatic cancer, which is highlighted in pink. It is tough to score these slides for disease severity because of different reasons. Firstly, they are massive in resolution, think billions of pixels, which each magnification level sometimes containing very different information. They can have stain variations that can confound models. And finding regions of interest within these images can sometimes feel like looking for a needle in a haystack. In addition to that, exhaustive labeling of these images is prohibitive as they require expert annotations and are time consuming. And this is where AI can add a lot of value. We can use it to reduce diagnosis variability through removing noise in this manual grading of diseases, which in turn enables us to better isolate the effect of treatment. And this is what the example on the left is demonstrating. It is for a disease called as NASH, or non-alcoholic steatohepatitis. It is a common chronic liver condition and one of the leading causes of liver transplant in the US. Another kind of use case for AI involves discovery of new biomarkers. And a biomarker is simply a measurable entity that is used to indicate the presence or severity of diseases. A simple example can be the ratio of cancerous cells to healthy cells. AI can provide quantitative, reproducible way to create such predictive biomarkers. Domain generalization is critical for enabling these applications that we discussed in the last slide. A domain can be any factor that has influence on the final image, ranging from patient characteristics to staining process and image acquisition techniques. It is difficult to predict the impact of a particular domain on the generalizability of an AI algorithm. And in some cases, such as for some kinds of historical healthcare data, the complete domain information may also not be available. This example serves to illustrate the point I was trying to make in the previous slide. These three images are all from the same dataset, which is TCGA lung adenocarcinoma. TCGA, or the Cancer Genome Atlas, is a large publicly available dataset. These images are scanned at the same scanning resolutions and scanned on the same scanner. Yet, as you can see, there is a wide variation in the image characteristics that aren't captured simply by the label domains. And not all images have all the domains available readily. ContriMix aims to solve this problem. It stands for content and attribute mixing. And the central idea is simple. It is to decouple the biological signal from technical noise in an image and then mix them to create synthetic images. So how do we perform this decoupling? It cannot be done for a single image since it is an underspecified problem. However, we can solve for content and attribute given a batch of images. ContriMix in particular solves this problem by extracting content and attribute tensors from input images. These tensors are then mixed together to generate synthetic images. These images, together with the original images, will be passed again to the content and attribute encoder to extract final output tensors. These output tensors are then used in the ContriMix laws to update the weights of the encoders. PyTorch, with off-the-shelf support for multi-GPU training, along with features like automatic mix precision, enables us to train faster and with larger batch sizes, which is beneficial for ContriMix training. Once this ContriMix encoder is trained, you can plug it into any downstream model training task as an augmentation module to generate synthetic images. And if you have labeled data, then you can also train ContriMix end-to-end with another laws such as cross entropy. And this is where the configurability of PyTorch comes in really handy. It is easy to reuse the same encoder multiple times within the same training loop, which is what we are doing in ContriMix, or to also have different kinds of strategies to mix patches through samplers. We can also generate attribute tensors from a new data set using an already trained ContriMix encoder, which makes it super easy to scale across new data sets from different hospitals, for example. So how does this technique perform in practice? We tried using it in a dataset called as Chameleon 17 Wilds. It's an open source benchmark for domain generalization by Stanford. Models in this dataset are trained on 3 hospitals, and then their performance is evaluated on out-of-distribution hospitals. Among the submissions following the official guidelines, ContriMix produces state-of-the-art OOD results on this dataset. We also observe some properties of these images which are generated by ContriMix. In this case, the content of the patch on the upper left was mixed with the attributes of 3 other patches on the right, which arise from different images. ContriMix was able to correctly transfer the color even when the input image is heavily affected by blur, as you can see in the 3rd column and top row. We also observe high correlation between content channels from ContriMix and biological signal, which can be something like cell nuclei or RBCs or red blood cells in these images. Additionally, visualizing content and attribute features through UMAP indicates that the attribute encoder captures the differences across patches from different hospitals, while the content encoder learns hospital invariant features. And all of this is happening without having any access to domain supervision during training. So in summary, ContriMix is a domain generalization technique developed for medical imaging, entirely using PyTorch. ContriMix offers advantages around not needing any domain IDs during training and being very easy to scale to new datasets. We have included the links for the code and the preprint here for the benefit of the broader ML plus healthcare community. And we plan to continue iterating on this and improving the code in the foreseeable future. Thank you. The main idea of ContriMix is that you do not need to specify domains at the time of training. So it can be something like hospitals. It can be the scanners that are used to scan these images. It can be some other characteristics. The idea is while training this model, it will itself learn to distinguish the biological characteristics, which is the content, from the technical noise, that is the attribute.